undergrowth, an encounter unfolds that seems hopelessly mismatched. A common cockroach, robust and quick, faces an iridescent green jewel wasp, Ampulex compressa, which is significantly smaller. The wasp doesn't aim for a killing blow. Instead, its first sting is swift and precise, targeting the thoracic ganglion. This initial injection isn't lethal, it's a temporary paralytic, specifically for the front legs, making the much larger prey manageable for the next, more delicate step. With the cockroach's primary defenses neutralized, the wasp begins the critical phase of its operation. It carefully maneuvers the roach's head, probing with its antenna. The stinger, now acting as a hypodermic needle and a sensory probe, searches for a specific entry point in the soft membrane of the cockroach's neck. It then delivers a second, much smaller dose of venom directly into the brain, targeting two specific clusters of neurons, the subesophageal ganglion and the protocerebrum. This second injection is not a paralytic. It is a masterclass in neurochemistry, a cocktail designed to achieve a single goal: the obliteration of self-preservation. The venom is a complex mixture, but its key components drastically reduce the levels of specific neurotransmitters, particularly octopamine. In insects, octopamine is crucial for initiating complex movements, like escaping from a threat. By blocking its function, the wasp doesn't paralyze the cockroach, it removes its desire to flee. The effect is almost immediate and deeply unsettling. The cockroach is not dead, not even fully paralyzed. It can walk, run, and jump if stimulated, but on its own, it does nothing. Its escape reflex has been surgically excised. The wasp can now approach, and instead of fleeing, the cockroach stands placidly. The wasp then clips the roach's antennae with its mandibles, not to disable it, but to create a handle. By grabbing one of the clipped antennae, the wasp begins to lead its victim. The cockroach, now a docile puppet, follows wherever it is guided, much like a dog on a leash. It walks at a normal pace, navigating obstacles as it normally would, but its destination is not its own. The wasp leads it back to a pre-selected burrow in the ground, a tomb prepared for a creature that is still very much alive. Once inside the burrow, the wasp's work is almost done. The cockroach is positioned, still passively awaiting its fate. The wasp then lays a single, white, oblong egg, carefully cementing it to the underside of the cockroach's abdomen, usually between its legs. This precise placement is not random. It ensures the hatching larva has immediate access to a soft part of the exoskeleton. The wasp then exits the burrow and begins to seal the entrance with pebbles and debris, entombing the living host. Why not just kill the cockroach? A dead organism begins to decay, becoming a hotbed for bacteria and fungi that would harm the delicate wasp larva. The venom-induced docility serves a vital purpose. It keeps the meal fresh. For several days, the cockroach remains in the burrow, alive and metabolically functional. It enters a state of hypometabolism, its bodily processes slowed down by the venom, preserving it for the horror to come. After about 3 days, the wasp egg hatches. The larva emerges and chews a small hole in the cockroach's leg to begin its first meal, feeding on the nutrient-rich hemolymph, the insect equivalent of blood. For the next 4 to 5 days, it feeds externally. Then, it burrows directly into the cockroach's abdomen. The cockroach is still alive as this happens. The larva knows, instinctively, to consume the organs in a specific order, saving the vital central nervous system for last to keep its host alive for as long as possible. A peculiar side effect of the brain venom is a period of intense, almost obsessive grooming before the cockroach is led away. For about 30 minutes post-sting, the cockroach meticulously cleans itself. Why would a neurotoxin meant to subdue an animal also induce hygiene? The leading hypothesis is that this is an antimicrobial strategy. By forcing the roach to clean its own cuticle, the wasp ensures its offspring's first meal is as free from harmful fungi and bacteria as possible. It makes its living pantry self-cleaning. Beyond octopamine, the venom also manipulates dopamine pathways. In most animals, dopamine is associated with motivation and reward. The wasp's venom appears to suppress dopamine signaling related to escape, but perhaps not other functions. Could it be that the cockroach's brain is being told that standing still is a rewarding behavior? This moves the manipulation from simply blocking an action to potentially rewriting the fundamental value system of the victim's brain. It's not just that it can't escape, it may not even register escape as a desirable outcome. After about a week of feeding, the wasp larva has consumed nearly all of the cockroach's internal tissues. All that remains is a hollow exoskeleton. Inside this macabre shell, the larva forms a cocoon and begins its pupation. The cockroach, which served as its prison, incubator, and first meal, now becomes its armor, protecting it as it transforms into an adult wasp. The victim literally becomes the cradle for its killer. Several weeks later, a fully formed adult jewel wasp chews its way out of the desiccated husk of the cockroach. It emerges from the burrow into the light, ready to begin the cycle anew. This intricate knowledge of neurosurgery, chemical pharmacology, and long-term food preservation is not learned. It is instinct, a complex behavioral sequence encoded into its DNA over millions of years of evolution. The wasp is a surgeon that never attended school, a puppet master acting out a genetic script of unimaginable cruelty and precision.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.